All right, guys, gals, it's 9 o'clock. Let's start our review session. This is going to have the same format as prior review sessions, so we'll do Q&A at the beginning, and then if you don't have any more questions for me, I'll just start tearing through the learning objectives. So, you have the floor. Any questions? So, do you want to go over the sarcomere and the fiber orientation, or do you want to do like excitation, contraction, coupling, and so the sarcomere and how it's related to the myofibrils and the myofilaments? Yeah. Okay, yes, let's do that. This will be something where it's easier for me to pull up a picture. So I'll go ahead and do that for you right now. Let's go to Top Hat. And this is one of those situations where if we had been able to cover gross anatomy of the muscular system before neurophysiology, it would have been better. What was that? Oh yeah, those A&P Flicks videos that I posted are way better than anything I could do for you in class, in my opinion. So let's see here, muscle anatomy, no sarcomeres there. So before I get to sarcomeres, let's start the big picture. The big picture is a muscle. A muscle is going to be attached to a bone by a tendon, wrapping all the way around that, interfacing the tendon to the bone, or the tendon to the muscle. We have a fascia, and the fascia merges with the membrane known as the epimysium. And the epimysium is going to take muscle cells and bundle them together in fascicles. And then if we look at one individual muscle cell within a fascicle, we can start to change our perspective and then focus on the inside of a muscle cell. Another name for a muscle cell is a muscle fiber. So here's an image of a muscle cell. And as we're looking at this individual muscle cell, we'll have the endomysium, which wraps around it to electrically insulate it from other muscle cells, and then we have the cell membrane of the muscle cell, known as the sarcolemma. And that sarcolemma will have invaginations, or tubes that go into it. I'll use green again. And those tubes are called T-tubules. In this figure, they're purple. And then right next to those T-tubules will be the sarcoplasmic reticulum, which is going to be the blue skeleton or the blue spider web of membranes in this cell diagram. And that sarcoplasmic reticulum is going to release calcium ions when stimulated electrically. Those calcium ions will then travel to the sarcomere. So let's go to our sarcomere. So as we get to the sarcomere, where is a good sarcomere slide? I know we talked about it. There we go. There's our sarcomere slide. So as we look at the sarcomere itself is going to receive those calcium ions, and it's made of two primary filaments, a thick filament and a thin filament. The thick filament is called myosin, and the thin filament is primarily actin. In this figure, the thick filament is the purple one, and the thin filament is the red or salmon-colored one. And then those calcium ions will get to the sarcomere and bind to the actin and allow for the thick and thin filaments to increase the amount of overlap. So that when we contract a muscle, we're going to take those thick and thin filaments and have them overlap more and cause the sarcomere itself to get shortened. Did that answer your question? they do not run through the thick as well. So those elastic filaments are going to go to the end of the thick filaments, and they also will go through the core of the thin filaments. Now, when we think of elastic filaments, the primary function of those elastic filaments will allow for the muscle to recoil to its resting length. If you stretch out the muscle too far, the elastic filaments pull it back in to resting length. There's another protein fiber at play here. The collagen fibers also resist overstretching and will allow for it to spring back to its resting length. Other questions?
I have a question not about this.
Absolutely.
I have a question. You said you asked me about like which organs are affected by the pituitary gland. Can you just go over that again?
So which organs are affected by growth hormone?
By like each hormone.
Oh, so let's talk about hormones of the pituitary gland. Sure thing. Do you want anterior pituitary, posterior, or both? Both, okay.
Just which organs, like everything else.
Sure thing. So, as we look at our pituitary gland, it's the anterior and the posterior combined together. And just to review, the anterior pituitary gland makes hormones. The posterior pituitary gland will store and release hormones. It doesn't actually synthesize hormones directly. So as we look at our anterior pituitary gland, let's go to the next slide where there's more room for you to write. The hormones of the anterior pituitary gland include follicle-stimulating hormone and luteinizing hormone. Those hormones primarily are going to affect the gonads, the ovaries, and the testicles for females and males. We also have thyroid-stimulating hormone, which affects the thyroid gland. We have adrenocorticotropic releasing hormone, or adrenocorticotropic hormone. ACTH is going to go to the cortex of the adrenal gland, or the outer circumference. Prolactin is going to go to the mammary gland, and then growth hormone goes all over the place. So that's the anterior pituitary. When we think of the posterior pituitary, we have antidiuretic hormone. That's going to primarily affect the kidneys. More than anything else, I want you to associate ADH with affecting the kidneys. Here's a funny story for you about ADH. Please don't raise your hand, but about 5 to 10% of the U.S. population really struggles with bedwetting, even into their teenage years. And there's also a lot of geriatric incontinence as well, nighttime incontinence, aka bedwetting. And to help combat this, oftentimes urologists or family doctors will prescribe an ADH tablet right before the individual goes to bed, or a skin patch with ADH. And the idea behind this is if you have a spike of ADH right before you go to bed, you're going to downregulate urine production while you're asleep, and you'll be less likely to have that nighttime incontinence. The other hormone of the posterior pituitary gland is oxytocin, known as the love hormone, oxytocin. If I had to give you a tissue that it affected, oxytocin affects smooth muscle tissue and nervous tissue. So it's really two tissue types, smooth muscle tissue and nervous tissue. So an organ perspective would be oxytocin affects the uterus, the mammary glands, and the brain. So uterine contractions, milk let down, and then feelings of attachment. So those are the 8 big hormones of the pituitary gland, both anterior and posterior. Any other questions? Mm-hmm. Did I write either on the answer sheet? Oh, that's a typo. That was because I was, absolutely. So when we think of myoglobin production is going to be upregulating the amount of a molecule that stores oxygen. So when we think of myoglobin, I want you to think of storing oxygen in the muscle. During aerobic exercise, long distance running, things that use your slow twitch oxidative fibers, you are going to burn through a lot of ATP. So you're going to need a lot of oxygen and glucose to make lots of ATP. So one of the modifications that occurs is as you do lots of long distance running, individuals that do lots of long distance running or aerobic exercise upregulate the amount of glycogen they store, and they upregulate the amount of myoglobin. By having more myoglobin and more glycogen, they can have more oxygen and sugar to use to make the ATP they need during aerobic exercise. Yes, so the hypothalamus has a little band, is connected to the anterior and posterior pituitary 2 different ways, depending on how you split it. So there's one chunk of tissue. That one chunk of tissue is known as the infundibulum. But when we look at the infundibulum, it has both neurons and blood capillaries in it. The blood capillaries of the infundibulum will connect the hypothalamus to the anterior pituitary, and then the neurons that go through the infundibulum will serve to connect the hypothalamus to the posterior pituitary gland. So when we think of anterior pituitary, it has a blood connection. Posterior pituitary has a neuronal connection. Any other questions? So just the excitation or excitation contraction coupling? Okay. I'll give you a simplified version of the neuromuscular junction, and then you let me know if it's too simple or if you want more detail. So when we think of the neuromuscular junction, we have an electrical signal traveling down a neuron. That electrical signal gets to the end of the neuron, and then all of a sudden, there's no more cell membrane. And we can't have the signal go from the neuron to the muscle because there's no physical connection between them. So we have to shift from an electrical signal to a chemical signal. So we go electric, 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 get to the end of the neuron, and then the end of the neuron releases a chemical that goes across the gap. That chemical goes across the gap, attaches to the muscle, and triggers another electric signal on the muscle. So that was a simplified explanation. Was that too simple, or do you want some more details? Okay. Well, I want to give some more details because it's hard for me to keep it that simple for you because I'm testing you at a higher level. So as that action potential is traveling down the somatic motor neuron, it's going to reach the end of the somatic motor neuron and calcium voltage-gated channels open up. When those calcium voltage-gated channels open up, they open and close in response to electric signals. So when the electricity gets to the end of the neuron, or the axon terminal, it opens up those calcium channels, and calcium rushes in to the end of the axon, or the axon terminal. Calcium ions are then going to attach to storage vesicles. And these storage vesicles are filled with the neurotransmitter acetylcholine. When calcium attaches to the acetylcholine storage vesicles, or as your textbook refers to them as synaptic vesicles, those vesicles will secrete the acetylcholine into the little space in between the muscle and the neuron, known as the synaptic cleft. Acetylcholine will diffuse across the synaptic cleft and bind to special receptors on the motor end plate of the muscle. Those special receptors are acetylcholine receptors. And when they have acetylcholine attach, the acetylcholine causes this protein receptor to open up, and it allows for potassium and sodium ions to move through the cell membrane. That movement of potassium and sodium ions triggers another action potential. So when we talked about electricity, I want to say 3 weeks ago now, in the body, we don't have electrons moving down a copper wire. The electricity in our body is going to be sodium and potassium ions moving across a membrane. So we start to move sodium and potassium ions, and as we start to move those sodium and potassium ions on the motor end plate of the muscle fiber, that triggers a new electric signal that then propagates across the cell membrane of the muscle, also known as the sarcolemma. So that was a more detailed explanation. Any other questions? So the 6 categories of joints? Sure thing. So as we look at our synovial joints, let's close up endocrine. Let's get to the joints.
Is there a special name for like joints?
To my knowledge, the joints are typically going to be named after the kind of movement they incur or some of the bones they are associated with. So it depends on the kind of joint. So let's see here. We have our classes of synovial joints. As we look at our classes of synovial joints, we have 6 total categories of synovial joints. The one that most people typically don't struggle with is the ball and socket joint. So you have a head of a long bone that's going to be associated with an indentation of either a flat bone or an irregular bone. The 2 classic examples of a ball and socket joint are the glenohumeral joint, where the head of the humerus sits into the glenoid cavity of the scapula. We're also going to have the head of the femur sitting in the acetabulum of the coxal bone. We're also going to have condylar joints, also known as ellipsoid joints. A condylar joint is going to be associated with the carpal bones, the interaction of our carpal and metacarpals. Our metacarpals and our phalanges. So as we look at these joints, these condylar joints are going to be a biaxial joint and allows us for the movement along 2 axes, or 2 planes of movement, which means you can bend your fingers and you can also crack your knuckles. As we look at a saddle joint, the saddle joint is named after the shape of a saddle. And when we look at our saddle joints, they're going to be associated with a metacarpal or a metatarsal interacting with a carpal or tarsal accordingly. So we have a metacarpal carpal relationship in the hand, and then in the foot we have a metatarsal tarsal relationship. These are also going to be biaxial joints that they allow for 2 degrees of movement, or we're going to have movement along 2 planes. We also have plane joints. These plane joints, to me, always remind me of tectonic plates, in that we have 2 pieces of bone sliding past each other. These plane joints are typically going to be associated with not a lot of movement. So we'll typically associate these as biaxial because we have those 2 planes, but there's a very little movement on those 2 planes. So when we think of the tarsals in our foot, they can flex and bend a little tiny bit. Or we think of the carpals of the hand, they can flex and bend a little tiny bit in 2 different directions. Another classic example of a plane joint is going to be a vertebrae to vertebrae interface. So if we think of in our vertebral column, between one vertebrae and another vertebrae, we're going to have a plane joint. And then the last 2 of our synovial joints are the hinge and the pivot joints. These joints typically, most students don't find that challenging. They're not quite as simple to comprehend as a ball and socket joint, but they aren't necessarily as rigorous as the condylar or the plane joints. So when we think of a hinge joint, this is going to be a monoaxial joint. Hinge joints are associated with a lot of bony structures that limit their movement, limit the degrees of freedom with that joint. When we look at a pivot joint, the pivot joint is going to be associated with a rotation of the bone, whereas a hinge joint is going to be associated with a flexion or extension of a joint. So when we look at the elbow, our elbow is a combination of both a hinge and a pivot joint. The hinge joint in the elbow is going to be the trochlear notch interfacing with the olecranon fossa. And then the pivot joint of the elbow is going to be the head of the radius pivoting within the ulna. We're also going to have another classic pivot joint, and that's the atlas and axis, or C1, C2 interface, that allows for us to turn our head from side to side and pivot. So that was the 6 joints in brief. Any other questions? In the front.
Like the differences in processes between immediate energy and short-term energy and long-term?
Absolutely. So we can, as we go through exercise, there are different ways we get energy for our muscles. We have the immediate source of energy, the intermediate, and the long-term source of energy. This is typically associated with like your first wind and your second wind as you're working out. So as we're looking at these sources of energy during exercise, when you first start to work out, you have a bunch of ATP floating around in the sarcoplasm of your muscle cells. And you can use that ATP right away to exercise, to move your muscles. And then you have a bunch of creatine a bunch of creatine phosphate and a bunch of myokinases, so that you can have the phosphagen system supply ATP. So initially, we're just going to use up latent ATP that's floating around in the muscle, and we're going to use up some latent glucose that's floating around in the muscle and generate ATP really quickly. And then after we've gone through that very quick source of ATP, we're going to go to a source of ATP that's a little bit harder to get to with the phosphagen system, with the creatine phosphate transfer system. I like to think of it as cash in your pocket, and then using a debit card, and then trying to liquidate a retirement account. So when you want to spend money, typically when most people, well, maybe not your generation, but when we think of an older generation, what the older generation wanted to spend money, the first thing they would do was pull the cash out of their pocket. That was the easiest way for them to spend money. And then that could be associated with just ATP floating around in the muscle cell. That's going to be the energy currency we use immediately or first, preferentially. After we're done with that energy use, we'll go to the next source of energy, or the next easiest way to power the muscle cell, which is going to be the phosphagen system. That can be analogous to making a withdrawal from your checking account or from using your debit card to make a withdrawal. It's a little bit more of a pain in the butt, but it's still not a huge deal. And then, after that, we're going to switch over to anaerobic respiration. Anaerobic respiration is something that our body does not prefer to do because it's very inefficient and doesn't give us a lot of ATP. And that's kind of like trying to take your 401k and liquidate it in order to have some spending money. Most people don't liquidate their 401ks because there's a lot of paperwork, it's a pain in the butt, and there's usually an early withdrawal penalty. In the same way, we don't like going through anaerobic respiration because there's a lot of extra steps, metabolically speaking, and there's a penalty associated with anaerobic respiration. Instead of getting over 30 ATP per glucose, we get 2 ATP per glucose. And then as we continue exercising, eventually we'll reach equilibrium. And that equilibrium point is going to be a balance of the balance out the rate that we can deliver oxygen to our muscles and the rate that we can deliver glucose to our muscles. And that's typically going to be dependent upon 2 things: your lung capacity and your blood circulation and overall body mass. So heavier people need to have more oxygen and more glucose delivered because they have to work harder to move their bodies in runs. So typically heavier people don't move as fast during long distance exercises. Lighter people don't have to move all that extra mass, so they can cause more movement with the same amount of ATP, and they typically will move faster. Any other questions? Absolutely. Let's talk about striations within a sarcomere. So how do the bands within a sarcomere influence the striations you see in muscle histology? Cancel presentation. Let's go to our striations. Where is that slide? because it's nice to have a picture up on the screen. So, as we're looking at the striations, just at a gross level, we have light bands and dark bands, alternating light, dark, that give us the striations of our skeletal muscle fibers. These alternating bands of light and dark correspond to thick and thin filaments. So when we look at the dark band, that dark band is going to be caused from an overlap of the thick and thin filaments. So there's twice as many filaments at the dark band area, so those dark bands are going to be darker. There's more material present to block the transmission of light. And then we have the light bands. The light bands are associated with a region that only has thin filaments. And because there's only thin filaments in the light band region, those thin filaments aren't capable of blocking as much light, so more light can get through, and we'll have the light band. Now, as we look at these bands and the structures associated with the bands, they have different names or different terms associated with them. The ones that I really like to emphasize, that I personally find to be the most important, are the M line and the Z disc. Because the M line's in the middle of the sarcomere, so it's the halfway point, and then a Z disc marks the beginning and ending of a sarcomere. Our Z discs are going to be in the center of our I band. So let's clear the screen here. So we have our I band here, which is known as the light band. And then in the center of an I band, we have a Z disc. From one Z disc to another Z disc is one sarcomere. So I find the Z discs to be very significant because they mark the beginning and ending of a sarcomere. And then in the middle of the sarcomere, we have an M line, M for middle. So this is one sarcomere, this horizontal rectangle I just traced out. So as we're looking at the M line, that M line, which is in the middle of the sarcomere, or in the middle of the dark band, is also going to be called the middle of the A band. So as we think of that dark region of the sarcomere, that dark region is also referred to as the A band. And then within the middle of the A band, where we have the thick filaments connecting to the M line, there's a little bit of an interface where we're not going to have overlap of the thin filaments. So there'll be a little clear space at the center of the sarcomere. That tiny clear space at the center of the sarcomere is referred to as the H band. And the chain of protein in the dead center of the H band is going to be called the M line. Do you have any other questions about the sarcomere? Okie dokie. So I really want to emphasize just the key point, like the really big take-homes of the sarcomere. The dark bands are associated with overlap of thick and thin. The light band is when there is no overlap and we have only thin filaments. The M line's in the middle. The Z disc marks the starting and ending of the sarcomere. Those are the really big take-homes. Any other questions? Yep.
But there's like, I can't remember which side it's on, but there's like 4 other ones, like the TRH, the CRH.
Oh yeah, those are typically going to be hormones associated with the hypothalamus. As a rule of thumb though, if I don't explain what the hormone does in typed text in the PowerPoint slides or in the presentations, I'm not going to ask you about it on the exam. Because there are just too many hormones out there. So if there's no written explanation, don't stress out about it.
Do we need to know like the names of them at least or no?
It wouldn't hurt to be familiar with the names, but don't focus on that. There's enough other things to focus on. Speaking of names though and hormones, For the hormones that we did cover in detail during class, you need to know the name and you need to know the abbreviation. So during the exam, I may ask you, what does ADH do? Or I might ask you, what does antidiuretic hormone do? Make sure you're able to use the abbreviation and the name. So you need to know the gland that makes the hormone. You need to know the full name of the hormone, the abbreviation of the hormone. You need to know the target of the hormone and the effect of the hormone on the target. What does that hormone do when it arrives at its destination? Any other questions? Sure. So that's going to be primarily of the hypothalamus and anterior pituitary gland, the hormones that cause other hormones to be released. So as we look at those hormones, the hypothalamus has lots and lots of hormones that it releases. And those hormones are then going to travel to the pituitary gland and cause the anterior pituitary to release other hormones. Of this list of hormones, or tropic hormones from the hypothalamus, the one that I really want you to focus on the most is the GnRH. That is a very key hormone. And the reason I want you to focus on that hormone the most is because when we get to the reproductive system next semester, we're going to spend a lot of time talking about gonadotropin releasing hormone, GnRH, for gonadotropin releasing hormone. And then when we get to the anterior pituitary gland, these three hormones are going to stimulate the release of other hormones in the body. These are tropic hormones. So these are hormones that cause the release of other hormones. So we're going to have follicle stimulating hormone and luteinizing hormone. Those hormones will travel to the gonads and trigger the release of other hormones. And particularly for the female reproductive system, I strongly emphasized during class that luteinizing hormone was the hormone that causes ovulation during the monthly cycle. There's also thyroid stimulating hormone, which travels to the thyroid gland and causes that thyroid gland to release T3 and T4, which is collectively referred to as thyroid hormone, a cocktail of triiodothyronine and thyroxine. The anterior pituitary gland also releases ACTH, adrenocorticotropic releasing hormone. ACTH will travel to the adrenal cortex and cause the adrenal cortex to release the glucocorticoids, those hormones from the adrenal cortex that help to regulate blood glucose levels and the stress response, notably cortisol. So those are the major tropic hormones, or the major hormones that cause the release of other hormones in our bodies. Any other questions? All right, let's pull up our learning objectives and get started on those, because we have about 34, so it's 34 after, we have about 15 minutes left. I specifically wanted to recover excitation, contraction, coupling, and cross-bridge cycles with you because that lecture recording was corrupted, and that was the one I got a lot of emails about. So I'm going to cover some muscle physiology with you, and then when I'm done covering that nitty-gritty portion of muscle physiology, if there's no further questions, we'll dive in and start talking about the learning objectives. So as we're looking at the neuromuscular junction, at that neuromuscular junction, as I said earlier, we'll have the action potential traveling down the cell. When that action potential gets to the end of the axon, or the axon terminal, it causes calcium ions to go in. Calcium is then going to bind to the storage vesicles filled with acetylcholine, also known as synaptic vesicles. And when calcium binds to those synaptic vesicles will fuse with the membrane and release acetylcholine, or ACTH. Acetylcholine will travel across the gap, or the synaptic cleft, to the motor end plate, and the little purple dots on that motor end plate represent acetylcholine receptors. When acetylcholine receptors have acetylcholine bind to it, they'll open up and allow for sodium to rush in and potassium to rush out. And as we have those ions moving in and out of the muscle cell, that changes the voltage across the membrane. And when we change the voltage across the membrane, we have two other protein channels, sodium voltage gated channels and potassium voltage gated channels, that open up in response to the change of the electrical voltage. And as those open up, they allow more sodium to go in, more potassium to rush out. That changes the voltage in a different part of the membrane and causes more voltage gated channels next door to open up. And then they allow ions to move in and out, and then next door, more voltage gated channels will open up, so on and so forth, and it just propagates down the membrane. As it's propagating down the membrane, that wave of electricity will move across the sarcolemma and eventually get to the transverse, or T-tubule. And it will start to depolarize the membrane on the inside of the T-tubule. When the membrane on the inside of the T-tubule is depolarized, question? Good question. What happens to the calcium that rushes into the axon terminal and causes acetylcholine to be released? Ultimately, that calcium is going to be processed and exositized. It will be released back into the extracellular fluid so it can be recycled and reused. It does not stay in the axon terminal forever. We just didn't emphasize that step because it's not one of the key components of this process. But you're thinking big picture, how do we maintain homeostasis? Because if we have calcium ions go into the cell, eventually those calcium ions need to leave the cell, or we're going to change ionic concentrations and lose homeostasis. Did that answer your question? Oh, so what happens to the calcium in the muscles? The calcium is actively transported into the muscle cell, and that calcium, while it is in the muscle cell, will be actively transported against its concentration gradient and stored in the sarcoplasmic reticulum. So we have calcium. Ultimately, we store calcium in our bones. And then from the bones, we'll put calcium into the blood. From the blood, we'll put calcium into the extracellular fluid, or interstitial fluid. And from there, we can put calcium into the neurons, or we can put calcium into the muscle cells. So we use calcium all over the place to regulate the electrical activity of our body. Any follow-up questions with calcium? Okie dokie. So where were we? Ah, yes, T-tubules. So our T-tubules are going to depolarize, or have that electric current travel across them. And as the electricity travels across the T-tubule, the terminal cisternate, or the ends of the sarcoplasmic reticulum, are going to also depolarize. And there's a special protein regulator that will open up in response to the change of voltage. As that regulator opens up, the sarcoplasmic reticulum releases calcium ions into the cytoplasm of the muscle cell. And the cytoplasm of the muscle cell is called the sarcoplasm. Once those calcium ions get into the sarcoplasm, they move through the sarcoplasm and eventually go to the sarcomere. While the calcium ions are at the sarcomere, they're going to attach to a special protein on the thin filament. That special protein on the thin filament is called troponin. And as calcium binds to troponin, so when these link up with each other, troponin is going to physically move and drag a secondary protein called tropomyosin with it. As tropomyosin is dragged out of the way, we have active sites on the actin exposed. And when the actin active sites, that's kind of a tongue twister, actin active sites are exposed, the myosin heads from the thick filament can now link to actin. In other words, we can now have the thick and thin filaments interact with each other. So at this point, we're going to start contracting. Another name for contraction is cross-bridge cycle. Most textbooks refer to the contraction phase as the cross-bridge cycle. And during this cross-bridge cycle, we're going to have, from my perspective, I always like to start at this step, where we have the myosin and actin interact with each other. So the myosin and actin attach to each other. As myosin and actin attach to each other, there's going to be a chemical bond linking them together. And then during this attachment phase, we're going to have this actin head, or this myosin head will bend backwards and pull the actin filament past the thick filament. So the myosin head bent backwards, pulled the actin filament past, and then they're stuck together. And they will remain stuck together until we have an ATP attach. When ATP attaches, the myosin head and the actin filaments will separate from each other. And this highly energized ATP is going to be used up, it will be hydrolyzed, and will recock the myosin head. So this step right here is commonly referred to as the power stroke, because we're using up the power that was put in the myosin head to pull actin past the myosin filament, and then a new ATP attaches and the myosin separates from actin. That new ATP is hydrolyzed and we recock the myosin head. And then we're allowed to repeat this process over and over and over again. So when we think of the cross-bridge cycle, there's 4 key steps. We have actin and myosin attach, we have the power stroke, we have a new ATP attach to separate them, and then we recock the myosin head, and it repeats over and over again. Every power stroke is going to cause the sarcomere to contract a couple microns. And this process will happen many hundreds of times to contract a single muscle. Now, that was the cross-bridge cycle, or contraction. Then we have relaxation. Ultimately, to relax a muscle, we need to stop electrically stimulating the muscle. When we stop electrifying the muscle relaxes. At a more nitty-gritty level, when we stop electrifying the muscle, we no longer have the T-tubules having the electric currents traveling through them. And if the T-tubules don't have electric currents traveling through them, the sarcoplasmic reticulum will no longer be dumping calcium into the sarcoplasm of the muscle cell. And instead of dumping calcium into the sarcoplasm, the sarcoplasmic reticulum will reabsorb calcium. And as the sarcoplasmic reticulum actively transports calcium out of the sarcoplasm into the SR, the calcium ions around the sarcomere go down and down. And as there's reduced sarcomere calcium concentrations, what we find is the opposite occurs on this slide. So the calcium will leave troponin. As calcium leaves troponin, tropomyosin goes back to its resting state and will block the active site. And then the myosin head can no longer interact with the active site of the actin because tropomyosin has reblocked that active site. And that's excitation, contraction, coupling, the whole process from start to finish. If you're looking for a good section of your textbook to read, I normally don't recommend you read your textbook, focus on lecture notes, but the section in your textbook on the neuromuscular junction, excitation, contraction, coupling, and then contraction and relaxation is particularly good. So that may be a good resource for you to look at. Question? Yes. So because troponin, I'm repeating this for the recording's sake. So because troponin returns to its resting state when calcium disengages, when troponin goes back to that resting state, it pulls tropomyosin back in front of the actin active sites, and myosin and actin can no longer have a cross-bridge cycle. We're down to 4 minutes. Any other questions? All right, I think we have time for one learning objective. I want to give you as much as possible out of this session. So let's go ahead and talk about rigor mortis. That's a common exam question. What is the physiological cause of rigor mortis? Which is related to this excitation, contraction, coupling, and the cross-bridge cycle. So when we think of rigor mortis, that is the stiffness of death, or the rigor associated with death. When somebody dies, they stop, having their heart stops pumping, they stop circulating blood, they stop delivering nutrients to their muscles, and the ATP concentration in the muscle starts to go down. As the muscle runs out of ATP, the muscle is no longer able to actively pump calcium into the sarcoplasmic reticulum, because that is an active process that uses up energy. So the calcium ions start to leak out of the sarcoplasmic reticulum, they bind to troponin, and initiate the cross-bridge cycle. But there's no more ATP, we're running out of ATP, so we can initiate the cross-bridge cycle, but we don't have any ATP to disengage the actin from the myosin. So we find that calcium leaks into the muscle cell, causes actin and myosin to come in contact with each other, and then there's no more ATP to cause them to disengage. All of the thick and thin filaments fuse together. All of the sarcomeres freeze in their current orientation, and at a gross level, this makes it so that you can no longer move that person. They become stiff. And to allow them to relax again, we need to cause the thick and thin filaments to disengage from each other. And typically, this is going to be associated as we have intracellular decomposition, which usually begins about 12 to 18 to maybe even 24 hours after death, depending on environmental conditions. Generally speaking, if it's warmer and more humid, rigor mortis will last for a shorter period of time. If it's cool and dry, rigor mortis will last for a longer period of time. Any other last-minute questions before we call it? All right. Well, in that case, happy studies. Please feel free to email me if you have any questions, or swing by my office hours today.